Good morning, everyone. Welcome back to our e-seminar series on Translational Biomedical Engineering. It's a great pleasure to host Professor Todd Hoare from McMaster University. Professor Hoare will talk about injectable hydrogels from discovery to translation today. But before we start his talk, I'd like to go over a few housekeeping notes. During the presentation, if you have any questions, please use the Ask a Question box. You can always use the chat box to communicate with the speaker and then other participants. But for us to be able to track your questions, please make sure that you type in your questions in the Ask a Question box. Also, I wanted to announce that our next speaker is Professor Hassan Olatak from University of Alberta. He will be talking about biomaterials to enable siRNA therapy for leukemic cancers. Please make sure that you mark your calendars. Also follow us on Twitter to get the most up-to-date information about these e-seminar series. Our Twitter handle is TransLBME. You can always email me and Human if you have any questions or comments or feedback about these seminar series as well. Last but not least, I'd like to thank our sponsor, Montreal Transmedtech Institute, who has been with us since almost the beginning of these e-seminar series. So with that, I'd like to invite our speaker, Professor Todd Hoare, who is the Canada Researcher in Engineered Smart Materials, the Tier 1 chair, and a professor in the Department of Chemical Engineering at McMaster University. His work focuses on designing hydrogels on all length scales, from the bulk to nano, for applications in diverse fields, including drug delivery, tissue engineering, agriculture, and biosensing, with a particular emphasis on developing rationally designed injectable in situ gelling hydrogels and micro-nano delivery vehicles. He is a co-editor of Chemical Engineering Journal, a fellow of International Union of Societies of Biomaterials Science and Engineering, a past NSERC E.W.R Steacie Memorial Fellow, and currently serves as the director of NSERC CREATE Training Program for controlled release leaders. Thank you very much, Professor Hoare, for accepting our invitation. The virtual floor is now yours.
All right. There's a lot of feedback on my end. I'm not sure if everyone else is hearing it.
Me too. Yeah.
I think you have two pages open on the same platform.
Sorry about this.
So, Professor, can you speak again?
There we go. Is that better?
That's much better.
Yes.
Perfect. Okay. Problem solved. Sorry about that. It's amazing. 2 years into a pandemic, it's the first time I've used this particular platform. So there you go. Thank you very much for the invitation to speak today, and for Mohsen and Human for organizing this series, which has been really good. I actually made my undergraduate students this summer listen to one virtual seminar every 2 weeks during their summer work term, and I read a lot of summaries from this series because I think it was very relevant to them and very relevant to a lot of people. So thank you for your effort in organizing it. So what I'd like to focus on today is some of our work on injectable hydrogels, and particularly those that we're really working on from a translational perspective. So hydrogels, just to make sure we're all on the same page, they're swollen networks of cross-linked water-soluble polymers. I just like to distinguish them briefly from a couple of other things that are related. So something like a cellulose sponge, for example, it can take up water, but it can only take up water because there's empty space. The cellulose does not actually soluble in the water solvent. So it's different in that way. Alternately, with a viscous polymer solution, there's no cross-links between the polymers.
Sorry, Todd.
So,
sorry. I think your slides are not moving and it's not on the full screen. So.
Okay.
And we lost Mohsen too.
Oh no. It's all falling apart. Okay. Let me try again. Okay. Is that any better?
If you go to full screen.
I am on full screen.
Maybe you can share the whole screen.
Sure. Just 1 second. Sorry about this.
Oh, no problem.
All works perfectly when we practice, and then when it actually happens, nothing works. Okay. Let's try this. Is that better?
Perfect. Perfect.
Okay. We're off. Sorry about that. So as I said, hydrogels are not like sponges because the sponge doesn't like the water, doesn't swell in the water, nor are they like viscous polymer solutions because there's no cross-link. So they will just keep expanding, and then dissolve. Whereas hydrogels will reach an equilibrium swelling point. Of course, hydrogels have been used for a lot of really important things in medicine, things like contact lenses, of course, spring to mind. Also personal care. I have had a couple of kids, so the diaper application was particularly important to me. Maybe less useful things like Jell-O. But in medicine, they've attracted a lot of interest because they have 4 key properties that are really beneficial in a medical application. Number one, their mechanics are very similar to soft tissues. So if we want to mimic something like a muscle or softer things, hydrogels have the ideal mechanics to do that. We can control their porosity by controlling how much the gel swells and also how many crosslinks are present. We can control the interfacial energy and maintain it low because of the high water content of the gel. So when this gel is placed in an aqueous environment, there's a very little difference between the free energy of the interface, which is already mostly water, and the bulk solvent. And if we want, we can make these with biomimetic chemistries, where the chemistry of the gel matches or is very close to those chemistries we find in native tissues. So the research objective in my lab is to basically try to innovate around hydrogels to address key biological requirements and limitations of the use of hydrogels in particular applications. And the one property I'll be focusing on today is the injectability of a hydrogel. We do that by learning how to control the chemistry, really thinking about structure property relationships, which I'll get into a bit more later, so we can design and characterize the morphology of the gel and relate that to how it behaves. Also looking at some new assembly techniques to put polymers together in different ways, either based on how we mix them or what they do after we mix them to get new types of properties out. And to validate my presence in an engineering department, we're also looking at some faster screening techniques and statistics and physical modeling to try to accelerate the identification of recipes or hydrogel designs that best meet application criteria. And then we design those for particular applications. That's in tissue engineering, a variety of drug delivery applications, biosensors, agriculture, anti-infective surfaces. And the particular type of hydrogel I'm going to focus on today are in situ gelling hydrogels. And the reason that these are so important is you can imagine if you're going to use a hydrogel biomedically, we have to get it inside the body. And you can just imagine, loading a piece of Jell-O into a syringe and trying to inject that. I'm not a particularly strong person, but even a strong person with a superhuman thumb is going to have trouble doing that because the yield stress of the hydrogel is so high. So this means that most hydrogels are either limited to use outside the body, so as topical applications, or they have to be surgically implanted. On the other hand, if we can actually create in situ gelling hydrogels, where we can go from low viscosity precursor polymers that can be injected and then gel inside the body, that removes that significant barrier to translation. So the idea behind these is that we start with precursor polymers. In our case, we look at covalently cross-linked gels, although there's a variety of physically cross-linked in situ gelling hydrogels that are also available that, based on physiological temperature or pH or other local stimuli can gel in situ in the body. But what we do is we functionalize different polymer precursors with different functional groups. So when we mix them together, those functional groups can react under physiological conditions and form a gel. And again, beyond just avoiding the need of surgical implantation, there are other advantages. If we're trying to deliver cells, if we do that in a very viscous polymer solution, for example, like a shear thinning hydrogel, a lot of stress is placed on the cells, and this results in relatively low cell viability at the end when we actually get it inside the body. Whereas here, the viscosity of the precursors are much less. Depending on the gelation time, which we can control, we can either gel it very quickly to localize the gel right where we put it, or we can give it a little bit of time to spread or wet interfaces, for example, preventing interfacial adhesions post-surgery. And also, depending on the geometry we bring these things together, we can make a lot of different structures through printing, through other types of processes. And I'll highlight a couple of these as we go on today. So we're designing in situ gelling functional hydrogels. There's really three things we have to think about. The first is the backbone chemistry. And that's basically the choice between what type of polymer are we using, particularly is it a naturally sourced polymer, where we're going to have some sort of typically native degradation pathway, and also typically some sort of native biological response, or a synthetic polymer, where we don't have necessarily those things, but we have a lot more control over the structure and a lot less concern about things like batch-to-batch variation, endotoxins, things like that, which can be very translationally critical. We think about the water affinity. How much do we want this gel to swell, or do we want it to swell? Do we actually want it to shrink? We can do either. Do we want to introduce a charge, or do we not? And do we want it to be responsive to the environment? And again, I'll pick up on that in a couple of slides. Then we can choose how we cross-link these backbone polymers together. So how long is the cross-linker? So how much space do we want between the two chains? What's the mechanism? So is it a physical mechanism or a chemical mechanism? And particularly if chemical, is it a degradable chemical mechanism? So does it degrade? If so, through what stimulus and at what rate in different biological environments? Specific to in situ gelling hydrogels, we also have to think about the rate of formation. So typically for an in situ gel, you want something from a few seconds to a few minutes. Faster than a few seconds, it gets hard to inject. And particularly a lot of the doctors we've talked to, they would actually prefer, a minute or two. If they need to inject a little bit, see what the back pressure is, inject a little bit more. So fast gelation is not always favorable. But if you leave it too long, you leave these low viscosity precursors too much time to diffuse away from the injection site, and that can really compromise the mechanical properties, the size, all that of the injected material. The last thing we can control is how many crosslinkers are there. So that's going to control the pore size and the elasticity of the mechanics of the gel, just as with conventional hydrogels. So we spend a lot of time thinking about this sort of triangle of structure, properties, and applications. And from a translational perspective, you can start, in our experience at least, either at the property box or the application box. So if we're starting at the property box, we can think of, okay, which key attributes or physical, mechanical, biological properties, whatever it is, do we need to actually make this hydrogel useful for something, even if we don't have a specific application in mind. In our case, I'll talk about injectability today. Then how do we design the structure of that gel in order to do that? And then once we see those properties, what types of applications do we think this will work for? Alternately, we can start at the application box where we have a very well-defined problem. And then we define the structure, the chemistry of the hydrogel to achieve a set of target properties we know we need to make that application work. So what I'm going to do today is going to tell you essentially two stories. One where we start in the property box and try to design injectable hydrogels and then find out ways to use them. The second where we start in the application box, where we have a very well-defined application to start with, and then we design the hydrogel to meet those application requirements. And the interesting thing, I think, is we kind of end up in the same place in both cases. So we'll talk about it as we go through. So for designing injectable hydrogels for biomedical applications, and we just wanted to solve the problem of injectability from a scientific perspective, the key question we asked is, well, what types of hydrogels should we make injectable? There are really two types we were really interested in. The first were protein or cell repellent hydrogels. And particularly protein repellent hydrogels, protein adhesion is well known to be the first step of inflammation, fibrosis, other sort of negative downstream effects. So we can minimize protein adsorption to a gel and also concurrently minimize cell adhesion. So particularly in the case of anti-infective surfaces, making sure bacteria do not adhere to the surface, so we cannot create biofilms nearly as easily. That would be beneficial. The other type of hydrogel we were really interested in are these switchable, or what we call smart hydrogels. These are hydrogels that when you change their physical environment in some way, And in biology, typically that's temperature, pH, or the reductive environment, although there's a lot of other types of stimuli that work. A hydrogel changes in some way, and it's typically a physical way. So it can change in size, it can change in porosity, change in refractive index. If it's a particle gel, it can change in how stable the colloidal gel is. So all these types of properties can change. When we apply the reverse stimulus or remove the stimulus, we can recover the initial state. So there's an element of reversibility involved as well. This has attracted a lot of attention in biomedical applications for things like on-off cell adhesion. So we can tune the cells from sticking to releasing, switching drug release kinetics. So we can turn drug release on and off to make release vehicles last longer and reduce side effects when drug is being delivered when it's not really needed. Or tuning the hydrophobicity, so tuning the interface, so how the body will respond to a particular implant or material over time. So the typical materials that we use for these, for antifouling, by far the most common is polyethylene glycol. And I think if you're on this call, it's very likely you're familiar with this. And this works based on two physical mechanisms. So one is sort of a brush-like physical repellency, where the high flexibility of this polymer basically sterically brushes, you almost think of it as a windshield wiper, the proteins from the surface as they come into adhere. There's also a strong hydrogen bonding effect this polymer has because of the ethers in the polymer backbone. So we get strong hydrogen bonding with water in solution, and as a result, you get these cages of water, which make it very difficult for a protein to come in and adhere to the substrate. But there's a key problem with this, or I wouldn't say problem, but limitation, and that this is a step growth polymer, so there's only two functional groups in it, one at each chain end. So I'm trying to make a hydrogel. I need to cross-link this. I basically need to use up virtually every functional group in the material in order to make a hydrogel. You can make star polymers and fancier morphologies to get around this, and people have done this with a lot of effectiveness, but it's more synthetically demanding. There's also some emerging concern about PEG-based antibodies, because PEG is in so many things that we are exposed to day to day. And the current rough estimate is about 40% of us have at least some form of a PEG antibody. And this is actually one of the reasons why people might have a response to the COVID vaccine. The Moderna and Pfizer vaccines, because they do have PEG stabilizers in the nanoparticles. So if we can avoid that antibody response, which is an emerging problem, again, controversial, but still something that we would like to avoid if possible, it would be good. There's also a lot of interest in making these temperature switchable polymers, and the key polymer there is polyanisopropyl acrylamide. And this is a polymer that exhibits what's called a lower critical solution temperature in aqueous solution. This is really interesting thermodynamic behavior driven by the interesting structure of this monomer, where we have a hydrophilic hydrogen bonding group and a hydrophobic group, the isopropyl group on the end. So as we heat up the temperature based on Gibbs free energy, the entropically driven transition, which is the self-assembly, the hydrophobic assembly of the isopropyl groups, begins to become more and more dominant over the enthalpic component, which is the hydrogen bonding. And we actually induce a phase separation, a physical phase transition. And again, this is completely reversible as we move the temperature up and down. So this has a lot of hugely beneficial properties, but the problem is this monomer is incredibly toxic. So if you look at the SDS of this monomer, basically if you can think of an adjective to put in front of the word toxic, it probably applies here. There's a huge regulatory challenge with actually translating this for in vivo use. There are a couple of acrylamide-based products approved in Europe and other jurisdictions, but to my knowledge, nothing in North America. The other polymer that's attracted a lot of interest recently is zwitterionic polymers for antifouling. What a zwitterionic polymer is again a polymer that has a negative and a positive charge in the same monomer repeat unit. And these are typically very close together. So you can see the monomer structures on the bottom right there. Typically, this is a quaternary ammonium group, which is a positive charge. And then 2 to 3 carbons away, there's a negative charge, which could be from a sulfate group, so a carboxyl group or a phosphate group most typically. Because these two charges are present, so they can, through polar interactions, order water, and because they're so close together, so if a protein is coming in, it doesn't really see a net charge. It sees, a net zero charge surface. These materials combine a ton of water. The estimate is 13 to 15 water molecules per monomer unit, and they also sort of mimic the native compositions of proteins. So in this context, they've been found to be very, very effective at minimizing protein and cell adhesion. But when we started this project, at least, there was no injectable hydrogel that's based on a zwitterionic polymer that would last any period of time. There was one reported, and it was a very short-lived polymer. So in terms of addressing some of these challenges, zodiac polymers we can use directly. There's no concern with actually translating these in the body. But as a substitute for PEG, which has these cross-linking challenges, and polynipam, which has these toxicity challenges, we really focused on this polymer called oligoethylene glycol methacrylate, or pegma. I'll say pegma for the rest of the presentation for obvious reasons as I trip over that. And this is a really interesting polymer because it has two key properties. One, the backbone is a methacrylate, so we can really easily prepare it through free radical or controlled radical polymerization strategies. But the side chain is polyethylene glycol. So what are the benefits of this? One, now that we can do free radical polymerization, it's really easy to functionalize the polymer. So we can just add another methacrylate, acrylate, to put whatever functional group we want inside this polymer. And this allows us to functionalize the polymer and also cross-link it and tune the cross-linking density by adding as many cross-linkable functional groups as we want. The other key benefit is depending on the length of the side chain, so the values of X and Y in the chemical diagram, we can dramatically change how this polymer behaves in solution. So if we have a short number of repeat units, so 2 repeat units, we get temperature responsive properties, very similar to those of polynipam. If we go longer, so a minimum of 5 to 6 units, but longer still, because there's commercial monomers available up to 40 units, we get highly protein repellent hydrophilic properties like PEG. But interestingly, there's been reports that there's no immune response to this brush PEG-like structure like there can be to the linear PEG. So there seems to be a differentiation between these things depending on how the PEG is presented to the whatever PEG antibodies may be present. So this polymer solves a lot of our synthetic challenges, but also allows us to really adapt the polymer for different uses. So what we're doing, I've really focused on, is the use of hydrozone chemistry to make injectable hydrogels based on these materials. And the reason we like hydrozone chemistry, which is the reaction between a hydrazide and an aldehyde or a ketone group, so I should mention that sometimes we can do this with the H group in the aldehyde replaced with another alkyl group to form a hydrozone bond. This chemistry really has two huge advantages. Number one is we can control the rate at which the crosslinks form from almost instantaneously to minutes, hours as we need. So this is perfect for injectability. We can control the gelation rate and thus basically customize it or tune it for different applications. The other benefit is that this is a hydrolyzable bond, but the hydrolysis happens pretty slowly. So it's on the order of days to weeks at acidic pH environments in the body. It can be months at neutral pH depending on the crosslink density. So again, we have a lever we can use to do environmental responsiveness, but also keep these gels around for a long time. Of course, these are synthetic polymers, so the backbone is not degradable. They're carbon-carbon backbones, whether we work with zodiac polymers or with the pegma-based polymers. So to make sure that they can be cleared over time, which is another key barrier, translational barrier, what we do is we keep the molecular weight very, very low, so below the kidney cutoff. So as the hydrozone bond degrades, we regenerate these starting materials, these oligomers, which have low molecular weights, and we have evidence that these are actually cleared through the kidney over time. So just very briefly, there's citations here if you want to know more details, but we've shown that these pegma hydrogels can have really highly adaptable properties. They can be thermoresponsive, they can be not. They can be transparent or not. We can tune the elastic modulus over 2 to 3 orders of magnitude. We can introduce thermal responses at particular temperatures, whatever we desire. And we can control the degradation time from very fast to very, very slow. Similarly, in the context of zodiac hydrogels, and this is using a sulfobutane monomer, so it's the sulfate, which has a negative charge, the quaternary ammonium that gives a positive charge. Functionalized in the same way with hydrazide and aldehyde groups and mixed with a double barrel syringe. What we end up with is, again, variety of tunable properties. But here, there's particularly interesting properties because the zodiac hydrogels, when we get them close enough, when they're far enough apart, we see a lot of water. That charge separation is so close between the positive and negative charges, it kind of blurs. We start to bring the polymers closer together in a cross-link structure, though. There's still discrete positive and negative charges in each monomer unit. And we get electrostatic interactions which actually can support the covalent hydrozone cross-linking. So what we found in this material is that we can get hydrogels to form at very, very low functional group contents, 5 mole % functionalizations or less, and much lower polymer concentrations, which means the water content of the resulting hydrogels can be higher, but still get very fast gelation and very long-lasting hydrogels. So this is a really interesting almost interpenetrating network, or dual cross-linked, maybe is a more accurate term, structures that we can get. In this case, we can, again, get very long-lasting hydrogels, months in neutral pH, but we can make that faster as well. Also, very good protein repellency, as you see in the bottom right, and pretty good bacterial repulsion without us doing anything else, as you see in the bottom left. So we've used these combinations of polymers for a variety of applications. So drug delivery, tissue engineering, cell delivery, nanostructured materials, fabrication. I'm going to show you a couple of these today. And really the benefits of this gel, again, it's degradable, integrate into clearable units. We have interfacial properties which are favorable, and based on how we bring these together, so how we mix the two precursor polymers, we can get different geometries. So we've made nanogels, microparticle gels, electrospun fibrous gels. So a variety of these types of structures that we can really control how these things are delivered into the body, how quickly they degrade, and how cells respond. So what I'm going to focus on now is just basically how we've been able to leverage a couple of these hydrogel technologies into more translatable technologies. So our strategy for commercializing these, obviously the first step is to patent it. So we have a patent on the pegma polymers, and we have a PCT patent current under examination for the zodiac hydrogels, as well as a couple of follow-up applications on particular applications. And of course, this is, I mean, if you're in the seminar, you already know this is the key thing that you have to do. If you don't have a patent, it's very difficult to have a subsequent conversations. But then we've really taken a two-pronged strategy building on that patent to actually get these into the market. I think the most interesting thing we've done that maybe not is less commonly done, to actually enter into a distribution agreement with Millipore Sigma. And I'm sure we've all ordered things from Sigma Aldrich. So if you go on Sigma Aldrich's website, they actually have a professor portal, and there's several professors around the world actually that offer products for sale through the Sigma website. So this is actually a very interesting contract. It's non-exclusive, so it doesn't stop you from doing anything else. So it's basically a materials transfer agreement. It's not a licensing agreement. And you're basically a vendor for Sigma Aldrich. You're not, there's no other encumbrance of the IP. The other interesting thing is that when they reorder it, if they reorder it, part of the agreement is the sales information, or who bought your product, is actually provided to you. So we just got reorder of our second batch a few months ago, and we got the sales information provided from the company. So it's actually very interesting to see who is buying this material, which, if you're trying to form a company or trying to translate it in other ways, might give you some clues as to what other people think it's useful for, which isn't always the same thing that you think the material is useful for. I would say this is quite a long time scale. It was over a year between when we started the process and when we actually had the materials online to sell. And you can see this is, Christmas is coming. Nothing says I love you like the gift of polyoegma functionalized with hydrazide groups. So, feel free. This is one of the product pages. We actually have 4 products for sale right now. And I won't tell you what the markup is on Sigma Aldrich, but suffice it to say, they should be doing quite well for themselves. But again, this has been a really interesting way to get our polymers in the hands of end users, which might find some interesting things to do with it, without giving up any sort of intellectual property rights or flexibility with other strategies. The second way we've been doing this is a bit more conventional, which is finding commercial partners that have interest in our technology to solve particular problems. And I would note that all 3 of these partnerships I'm going to describe are actually seated at conferences, so that people from the companies were in the audience. They heard me talk, or one of my students talk about these hydrogels, and we connected after to see whether there's something we could do. So the first partnership, and probably the one that's the furthest along of this, is the use of these hydrogels for viscosupplementation. And this is to address a huge problem in the context of osteoarthritis. So osteoarthritis, as many of you know, a huge issue in joints where synovial fluid breaks down, so the lubrication of the joint, and or the cartilage at that joint, which again provides the cushioning as we provide impact, starts to break down. So over time, you get basically bone-on-bone, non-lubricated action, which can cause immense pain and usually ultimately leads to a knee replacement. This is a particularly difficult problem for younger people who have this, and particularly athletes, because you can't replace your knee that many times. Typical knee replacement time frame is 10 to 15 years. So it's very difficult to choose to get a knee replacement when you're quite young. So one solution that's been proposed to this is this idea of viscosupplementation. So instead of just letting things go or just trying to treat the inflammation, adding a material that can supplement or replace the synovial fluid or the cartilage at the joint and provide the cushioning and the lubrication. And there's several products on the market right now that claim to do this at least, and almost all of them are based on hyaluronic acid. And that makes sense in a way. There's an inherent anti-inflammatory property of hyaluronic acid and hyaluronic acid in native synovial fluid. The problem is that none of these are actually cross-linked hyaluronic acid. They're all just high molecular weight hyaluronic acid. So these viscous solutions, which, as I said on the first slide, over time, particularly in an infinitely diluting environment like the body, are going to just dissolve away. There's also a lot of enzymes which are actually upregulated during inflammation, which can specifically degrade hyaluronic acid. And the best guess now is that if you pay for this $800 injection, and it can be more in some jurisdictions, that's sort of the average cost, you're probably going to have the material in your joint for 2 to 3 days, and that's about it. And sort of more macroscopic studies have shown that really there's no huge benefit to these injections versus a placebo. So you're paying a lot of money putting a needle in your knee for something that, to be honest, usually doesn't work. So we've been really active with our partner Trillium Meditech on this to develop these formulations specific to the osteoarthritis area. And our lead formulation is right now called VetSen, and I'll tell you why it's vet in a second. But this is actually one of the zodiac polymers, which is gelled with a mix of aldehyde and ketone groups. The reason we chose this is aldehydes, you might be thinking, well, aldehydes can react with proteins. They can through shift-base interactions. Is that a concern? So we're trying to dilute this concern by mixing the fast gelling aldehyde, which allow us to do in situ gelation, with the much slower but still eventually gelling ketone groups, which do not have that type of possible cross-reactivity with proteins. You can see we can replace the, very well simulate the viscosity of hyaluronic-based gels. And also with this material, because it's in situ gelling, going from low viscosity precursors to a gel, we actually get not to a cartilage mechanics, I wouldn't claim that, but we're on the same order of magnitude as cartilage mechanics. So we could choose to go either supplement the synovial fluid or the cartilage using these types of formulations. The really exciting result, though, was the lubrication result. So this is actually, if you compare the hyaluronic acid-based gels, which again degrade very quickly, they're very shear thinning, with our cross-linkable zodiac polymer based on hydrazide and aldehyde/ketone groups, you see whether we're walking or we're running, we get between 3 to 5 times higher lubricity. So this is basically lower energy losses associated with the movement, which relate to patient comfort. It also means we can get away with a bit stiffer material and still not lose that patient comfort. We've done quite a few studies in vitro biodegradation where we show in chondrocyte spent media. So this is a media that has been recovered from chondrocytes, so that's the enzymes that would be present natively. We see our material doesn't degrade really at all over 30 days, whereas hyaluronic acid disappears almost entirely in 2 or 3 days, which is not inconsistent with the literature. We see this in animal tests. This is in the rabbit knee. You see that the gel does break apart, which we didn't anticipate, honestly, but we think could actually be another benefit because now these particles can also move past each other, and it might actually help with lubrication even more. And we don't see the inflammation that might be the red flag with that approach. We've also had this in a dog joint, and you see no inflammation or problematic morphology 4 weeks post-injection, nor is there any change in the protein content in the synovial fluid. So we're now at the point of, on our lead formulations, starting some rabbit models with osteoarthritis to prove this works. And our first target is actually the vet market. I did not know this, but there's about $150 million market for osteoarthritis viscosupplementation injections in horses and dogs in North America. So you want the family pet to be able to walk, and you want your racehorse to compete a little bit longer and get more money from it. So this is actually our first target. It's a much more regulatory light area. It's basically proving it's safe and then post-market regulation and monitoring before we actually go into a human application. We've also seen that these materials, particularly the zodiac materials, are really good for delivering cells. And we didn't anticipate this because a lot of these protein repellent materials, they're also cell repellent. And as such, cells, you can keep them happy in the gels for a few days. But typically, because they can't adhere well, over time, they get a bit stressed out and they start to die. That doesn't seem to be the case with these zodiac hydrogels. And we're still trying to figure out why. But you can see depending on the number of functional groups we have, so the percentage in slide D there corresponds to the mole percent of hydrazide or aldehyde functional groups in our material, our precursor materials, we not only get viable cells, but we get actually proliferation inside our matrix. If we use a material that degrades on a reasonable period, so 7 to 14 days, we've actually seen in the top image, it's a little bit hard to see visually, but the cells release, they can stick to the bottom of a plate and they start proliferating themselves. So really excited about this potential to combine maybe viscosupplementation and actual cell delivery to help not only address the problem, but also actively repair the problem over time. But of course, that's a longer term goal. Second problem I want to talk about is the idea of printable platforms for drug discovery. And again, we had no idea this was a problem until we were in a conference and someone started talking to us about it. And this is a problem for small molecule drug discovery, in that these are typically done through high throughput screens with thousands, tens of thousands of molecules to identify hits of molecules that inhibit the performance or the turnover of a particular enzyme you're trying to inhibit. The problem is that up to 95% of the hits that are identified in this first stage high throughput solution screening actually turn out to be what's called a promiscuous or non-specific inhibitor. That it's not a specific binding event that causes the inhibition. It's the fact that the molecule, the small molecule target, actually aggregates under the conditions that aggregate physically blocks that enzyme binding site. But of course, it would also block every enzyme binding site. So in terms of making a drug, it's not useful. This has been highlighted in a lot of editorials of late. So we're asked the question, well, can your hydrogel actually address this? Because we know with the in situ gelation, depending how we mix it, maybe it's printable on surfaces. We know we can control the porosity by the density of the polymers, by how many functional groups are present. And we know that it has good non-specific absorption. So it should be good for the selectivity of the process. So what we did was to try to print these hydrogels onto basically nitrocellulose paper sheets. And the benefit of doing this versus a traditional solution assay is that we can really reduce the volume that we need in order to assess drugs of particular concentrations. So for drugs that are potentially very interesting, but are isolates from bacteria or plants, very hard to get large volumes of them. This can reduce the screening requirements, so the amount of these materials that are required to do a comprehensive screen. So what we did is we actually did drop-on-demand solenoid printing, where we sequentially print the hydrazide polymer and the aldehyde polymer, usually the aldehyde first, simply because the nitrocellulose platform or any paper platform, there's always a few aldehyde groups around. So if you dry the aldehyde polymer, you can get a little bit of cross-linking to help to anchor that polymer, and as a result, the hydrogel formed by the Hydrozone click chemistry to that surface. What you can see in the image is if we just print non-cross-linkable hydrogels, they rinse right off the paper. But if we sequentially print our aldehyde and then hydrazide polymer, you don't see that bumpy morphology in nitrocellulose anymore. We've created a gel layer on the surface. We also showed that enzymes can be co-printed without any significant loss of activity. And interestingly, this can be just sitting on the bench at room temperature we've tracked up to 3 months, where there's no significant activity loss. And we think this is just because these hydrogels are very, very water binding. It keeps that hydrated environment around the enzyme. So what we did is we printed a enzyme that was relevant to drug screening and inside our hydrogels, and then we applied what was a known enzyme inhibitor, so something that was actually a drug for this enzyme, so a true enzyme inhibitor that can actually bind, and also something that gave a positive result in a solution assay, but is a known promiscuous or aggregative inhibitor. What you can see in the solution assay in the bottom with the blue points, it looks like both of these work, right? It looks like both of these can actually inhibit the enzyme and both could be drug candidates. But for the enzyme printed inside our gel, you can see that only the one that is actually a true inhibitor that can actually diffuse through our gel, bind to that hydrated enzyme inside the gel, it gets the exact same LC50 value as we get in solution, whereas the one that aggregates and is thus too big to get into the pore structure of our gel and thus can't no longer interact with the enzyme, we see no signal, no inhibition at all. So we think this has a lot of power to actually minimize the number of false leads that are pursued in the much more expensive second phase of drug discovery. And we're in talks with a drug company to perhaps try to implement this in some of those screening strategies. We've also sort of tried to expand this to see, well, if we can encapsulate enzymes, can we encapsulate other things? And this is an example of a rolling circle amplification, or RCP product, which is just a really long strand of repeated DNA sequences. So up to a megadalton molecular weight of the same DNA sequence over and over and over again. What we saw when we print this in our hydrogel using the exact same printing strategy is that if our sequence that we're trying to the probe that we're trying to detect has a lot of base pairs, we no longer can detect it. So there's a selectivity based on the porosity of the gel, where these larger base pairs, which can be competing species, can't actually get in. On the other hand, if we're trying to detect very short base pairs, what we get is about a 2-fold increase in signal to noise. And what we think the reason is, this can easily diffuse into the gel, so we can still interact with this rolling circle amplification complementary DNA sequence inside the gel. But the hydrogen bonding interactions with the gel can help to stabilize the duplex that's formed and actually create a stable binding event, which in solution with all the competing hydrogen, water, and the mobility competition that's involved, creating other entropic barriers to this binding event from happening, creates a lot more challenges doing this in solution. So we're really excited about this approach, actually being able to detect lower, much smaller sequences of nucleic acids in solutions and in complex solutions, taking advantage of that non-fouling property. The last partnership I want to talk about is in 3D bioprinting. And again, many of you on this call, I know Human and most of them are really involved in this area. There's a lot of interest to do 3D bioprinting of hydrogels, both for functional implants as well as for in vitro drug screening and mechanistic studies. Right now, I think it's fair to say that a key barrier in this area is the availability of inks. So you can either use a shear thinning ink, which you take a hydrogel and that shear thins, you can extrude it through the needle and get a hydrogel at the other end, but you're limited in mechanics because you have to still be able to shear thin it enough so that it moves. You can do coagulation bath printing with things like calcium alginate, but then you're really limited in chemistries because almost everything you has to have at least calcium and alginate as components of the ink, if not the only component. Or you take to photopolymerization. And again, for some cells, that's totally fine. But for other cells, you need to expose the cells to UV light, particular wavelengths, or over particular times can be problematic. So injectable hydrogels are an obvious solution to this, but there's a huge problem in that mixing in a continuous process over a longer period of time with these gels is really challenging because depending how you mix these things together, there's a very high risk of the print head clogging. So what we've done is a couple of strategies. The first is we designed a coaxial 4D printer, and this is kind of a custom-built thing with components off the shelf, which we could use to co-deliver these reactive functional precursor polymers and create printable hydrogels. So what the tip looks like is basically a smaller needle in which we print the hydrazide component typically, and cells if we want to print cells, then an outer needle where we deliver the aldehyde component. At the interface, it isn't completely blunt. There's just a small gap between them. There's a small chance to mix, but it's essentially just a coaxial flow, and thus diffusion of these low molecular weight precursor polymers that are driving mixing in this case. But in this case, we can actually print hydrogels fairly effectively. You see in the videos, you're printing lines, droplets. We create combinations thereof to create some interesting grids. The other thing we found is if we print on hydrophobic paper, and actually create almost discoid-like structures, which we can almost match the size and morphology of red blood cells. And this is something that weren't expecting, and we're now quite interested in trying to pursue. Just to give you an idea, these are not fantastic images. This is still kind of early in this project, but you can see that cells can be printed. And particularly if we add in collagen to give cells adhesion sites, pegma is not great for printing cells in terms of the lack of adhesion sites that are present. We see better cell morphologies and a bit more proliferation in some of our longer days, which I'm not showing. But the key point is here, we can print viable cells in patterns, and we're now starting a collaboration with Aspect Biosystems, which is a really unique and interesting microfluidic 3D printer, to try to control the mixing of these polymer precursors much better, and thus get much better mechanics as well as integrity of the final printed structures. The other thing we've been looking at is to try to build more 3D structures with this material is to look at the freeform reversible embedding of suspended hydrogels, a fresh bioprinting strategy, which is where these hydrogels are printed into a gelatin microparticle bath. This is really pioneered by Adam Feinberg at Carnegie Mellon. And this helps to physically support the gel as you build it. So it's much easier to build 3D structures in these supported bioprinting strategies. You can see that we've been able to do this successfully using two approaches. One is where we have the hydrazide polymer and cells inside the needle, and then we print those into the mixture of the gelatin particles and our aldehyde polymers. So in this case, the cross-linking happens as the aldehyde polymer diffuses into the printed hydrazide solution, which is sort of held in place by the gelatin particles for long enough for that cross-linking to happen. Again, you see a little bit of anisotropic swelling events here, which we're trying to address, but it does work to build fairly high and maintained structure hydrogels that are printed. The other thing that we can do, which is maybe even more interesting to us right now, is if we use ketone functionalized polymers instead of hydrazide polymers, where now the gelation time can be tens of minutes, this bath can actually keep those precursor solutions in place long enough that tens of minutes gelation time isn't a problem. Because if you're doing freeform printing, that's obviously going to be a problem. You'll just create a big puddle or blob at the bottom of your 3D printing well. So in this case, because the materials are pre-mixed, we get much better cross-linking, we get much better printing resolution, and we've also seen we get much better cell viability. So you can see every green dot in the image on the left is a cell. We can print at extremely high cell densities and with a quite good resolution on the printed features. And we get a lot of actin staining when we try to do that on the images on the right to indicate that cells actually are adhering well to this platform. And this is actually with one of our zodiac printed platforms. So again, we're looking to see how far we can push this, what structures we can get, what good of resolution we can get, and what applications we can ultimately do to hopefully engage partners in this as well. So up to now, I've been talking basically about we tried to solve a technical problem, and here's sort of the things that dropped out when we tried to do that. For the last few minutes, I want to actually flip it around to describe a technical challenge we were faced with and how we actually designed hydrogels specifically for that technical challenge. The technical challenge we were faced with is antipsychotic drug delivery to the brain. And as many of you know, drug delivery to the brain is really, really hard. Not only because of the blood-brain barrier, but also because of all the other barriers in terms of first-pass metabolism, degradation of the gut microbiota for orally administered drugs, which make it hard for the drug to even get to the brain to have a chance to get through the blood-brain barrier. This results in a very low fraction, 5% is probably very generous for most drugs, of the percentage that actually gets to the brain. We had collaborators with Ram Mishra's lab here at McMaster that have really been interested in intranasal delivery of pharmaceuticals to the brain, which is a really different route in that there's multiple direct pathways through the olfactory bulb or the branches of the trigeminal nerve, which as long as you can get the drug through the nasal epithelium, which in this case is the key barrier, the drug can travel along those pathways directly to the brain, bypassing all these other barriers that are present in terms of first-pass metabolism, gut microbiota, and the blood-brain barrier. This is also a relatively easy way for patients to administer drug if we can design a sprayable formulation to do this. And as a result, there's a potential to get fewer side effects at lower doses. In particular for antipsychotic drugs, some of the side effects are fairly unpleasant. So avoiding side effects will really help with patient compliance, which is a huge issue in this area. There's also faster onset because there's fewer barriers to the diffusion to the brain, but concurrently also faster offset. So we need to be able to still do controlled release so that the patients don't have to administer their drug more frequently. So here are really the four key design criteria we're presented with. It has to be sprayable. We have to also retain it in the nose as a result of the spray. So we really were focusing on mucoadhesive materials to do this. We have to have some mechanism to get the drug across the nasal epithelium because that's a key biological barrier in this case, not the blood-brain barrier. So for that, we wanted to use nanoparticles, which have known active transport mechanisms across the nasal epithelium. And we needed slow release to sustain that therapeutic effect. So what we decided to design is what we call a nanoparticle network hydrogel, which is comprised of two components here. One is carboxymethylchitosan, which is just a soluble derivative of chitosan, but a material that has these amine groups, which we can use for cross-linking and also have been found to be very beneficial for mucoadhesion. We combine those with starch nanoparticles. These are particles that were made by one of our research partners, Ecosynthetics, usually for paper coatings and wood laminate adhesives, not for biology. But it had extremely low particle sizes, and as a result, we found them to be extremely good at penetrating through tight junctions and getting through tissues that are hard to penetrate through, such as the nasal epithelium. They're also enzymatically degradable, clearable. They're based on starch, so that's easy, good for regulatory. And we can easily oxidize a few of these repeat units to form aldehyde groups. So if we mix the amine-containing carboxymethylchitosan and the aldehyde-containing starch nanoparticles through shift-base interactions, we can get a gel. And we found in vitro that this could be sprayed through a mucosal atomization device. So this is a commercial device that is currently used for sprayable treatments to the nose. Particularly at the lower carboxymethylchitosan concentrations, we can actually spray a distance that's relevant and also an area that's relevant to the human nasal passage, which we're really excited about. We can also get gelation happen in less than 5 minutes, which is a good time frame for a thin film hydrogel sprayed to the nose. We also can get tunable swelling and degradation, and in this case, between 2 to 7 days. And that's appropriate because it's very similar to the rate of mucosal turnover in the nose anyway. So beyond that, it's really not useful to have the material sticking around. And you'll see particularly the 35 weight percent SMP formulations with 2 and 4 weight percent chitosan, they didn't swell a lot, so we wouldn't anticipate it closing the nasal passage, but they also had the ability to stick around for that time period. So those are the ones we really focused on. And to validate the in vivo performance of this, we actually did a schizophrenic model by inducing schizophrenia-like symptoms, particularly social withdrawal, using a molecule called MK801, which you administer through IP injection. And then using this tripeptide molecule, PEOPA, which was developed in our collaborators group, which is known to prevent and reverse behavioral abnormalities in preclinical animal models. So in this case, we're delivering a peptide intranasally using our material. And we looked at this through a social interaction paradigm where we put 2 rats who had never met each other before inside an arena, and we'll just let them interact. So if they were under schizophrenic symptoms, social withdrawal is one of the symptoms of that, they would interact less in terms of sniffing, in terms of fighting, in terms of grooming, whatever it is they're doing. Whereas if the schizophrenia can be solved by our material, they will interact more. What we saw experimentally is if we don't treat these animals for the 5-minute time we view these in the arena, we call it. Sounds like a fight, but it's really not in the arena. For 2 of the 5 minutes or so, this is measured by a blinded observer, the rats were judged to be inactive interactions. If we deliver our vehicle alone without drug, or the drug alone without the knockdown, without inducing the schizophrenia symptoms, we see the exact same interaction time. Once we induce the symptoms, though, if we just treat with artificial cerebral spinal fluid, we see about 1/3 of that interaction time now. So the huge change in the amount of time the rats are actually interacting with each other. If we administer PEOPA, the drug, through the nose just alone, after 1 hour, we recover most of the behavior. After 24 hours, there's no residual benefit at all. So if we deliver the same dose of drug using our hydrogel, and in particular, the lower dose carboxymethylchitosan hydrogel, which again has a much better spray property, we see we can relieve the schizophrenia symptoms entirely for 72 hours and with still some residual positive effects out to 5 days. This is a huge, huge difference where, for patients that may not be as compliant with their medications, they have to do 1 single nasal spray and get beneficial effects for, at least half a week. That's a really beneficial development. And we've seen when we tracked the biodistribution of these materials that a lot of it gets stuck in the nose and stays stuck in the nose for, the other 3 days that we looked at it. But a significant portion, not a ton, but a significant portion of the starch nanoparticles actually do transport into the brain. So we think that the benefits that we're seeing are a combination of some of the starch nanoparticles as the gel degrades because of the reversibility of the shift-based crosslinks, actually transporting actively through the nasal epithelium and transporting to the brain through those pathways I showed you. But also some of the nanoparticles actually getting taken up in the nasal epithelium and basically acting as controlled release vehicles to release the protein over time. The protein itself can transfer through the brain since there aren't any of those other barriers present once it's past the nasal epithelium. So we initially designed this as a solution to a specific problem, the delivery of peptides to the brain. But we've now been thinking of this as a bit more of a platform technology because it's really easy to functionalize these starch nanoparticles. The way they're made is through an extrusion-based process, so the starch is amorphous. So we can do almost any chemistry we want on these. So we're looking at this for a variety of applications. In this case, levodopa is our target, which is the primary current treatment for Parkinson's disease. And it has very low bioavailability because there's a lot of degradation in the gut microbiota. It's also one of these catechol-like molecules, which have 2 hydroxyl groups adjacent to each other on an aromatic group, very susceptible to oxidation to ketones, which not only make the drug not work, but can also induce toxicity. So what we've been doing in this case is keeping the aldehydes so we can get the gel forming, but also introducing these phenylboronic acid groups, which are known to bind reversibly to cis-diol groups, such as those on L-DOPA, to stabilize them and also to control their release. When we induce schizophrenia-like symptoms, in this case, these little C. elegans that you see moving around, wiggling in the microfluidic channels. There's an electric field that's driving them to move. You can see if we apply our gel and treat them with the gel, they sort of have that normal coordinated faster motion that you would expect with an untreated worm. So that means the schizophrenia symptoms of the model are being resolved. Whereas on the right, this is with no treatment, just induction of the symptoms, but not treated in any way. You see sort of slower and very uncoordinated movement consistent with Parkinson's disease. And we can maintain this for 48 hours, or at least 48 hours. So if we apply the drug alone at the same dose, it actually stops working after 5 hours. And we actually get a little bit of toxicity to the worms as the keynote forms. So again, by adding different functional groups, we're really hoping to customize these delivery vehicles for different types of drugs we want to transport to the brain and help address many of these mental health issues. And again, we're in conversations with a pharmaceutical partner to actually test this on one of their peptide drugs that they want delivered. So last slide. I just want to, many people in this series have been, they have products on the market that are impacting patients now. We have a bunch of products that are under development and in advanced animal models hoping to get to patients. So you can take these lessons learned with a grain of salt they're probably worth. But these are the things that I've sort of taken home from our experiences thus far, trying to translate our research. The first is what we think is useful is often not what other people think are useful. So when we developed the injectable hydrogels, we were really thinking, for sure, this is for drug delivery. For drug delivery of proteins, this is a great solution. You can see all three applications that have actually attracted industrial interest have nothing to do with drug delivery of proteins. In fact, one of them had to do with trapping proteins so they were never released. So I think it's really important to be really flexible, to talk to as many people as possible about the technologies you have, and be really responsive to the polls and their ideas on how their technology could be used. Second thing is a lot of our materials are platform technologies where they can be adapted for different applications. And for our injectable hydrogels, obviously, we designed it as a platform technology and then tried to find uses. But the real thing we learned there is a platform technology is great, but it's not really useful if you can't show the partner you can actually solve their problem with the platform technology and adjust the platform technology to solve their problem. I think it's really important to understand what those problems are and to show that your platform technology can actually impact that issue. Third, we've had a lot of success engaging with our university's media office to get our story out in the press. And again, we got an article in the Hamilton Spectator for the last article, the intranasal drug delivery that I just talked about. Just say, okay, great, Hamilton Spectator. I'm guessing none of you read the Hamilton Spectator. Who would have seen it? But it also goes to these trade publications, and both of the leads that we've gotten for companies interested in that intranasal spray have been from people who've seen the press release from McMaster in a trade publication and had some interest to talk to us. Fourth, when in doubt, file a provisional patent before a public disclosure. We've gotten in trouble with this. Some of our zodiac hydrogels might not actually be able to be used for some applications because we talked about it before we filed it, before we really realized why it was actually so useful. So when in doubt, file before you chat. And third, as you are on the seminar series, learning about entrepreneurship and technology transfer, it's really a worthwhile investment of your time, no matter where you are in the student, professor, industry area. And just before I wrap up, just a quick pitch here. We have started just this month a program that if you're interested in drug delivery, will really hopefully help you to do that. So the NSERC CREATE Training Program for Controlled Release Leaders, which I lead together with 10 other professors in the sort of GTA to Montreal corridor. And this is really an industry integrated program where you get a chance to really learn design thinking skills to design controlled release, not only for medical applications, but also for agriculture, food science, other type of applications. So if you are a student looking for a graduate position or a postdoctoral position, please check out our website, follow us on Twitter, find out more, and feel free to contact me for more information. We're actively recruiting now for May or September 2022 starts. So in conclusion, I'd just like to thank the students that did all this work and my collaborators, particularly Ram Mishra and Carlos Felipe, and our funding partners. That's our last pre-COVID photo when we could actually stand less than 2 meters away from each other. But it's a great group to work with. I'd be happy to take any questions you might have.
Thank you so much, Professor Hoare, for this great presentation. You brought up lots of applications, and we got several questions. You can stop sharing, and okay. So we have several questions from audience here. I don't know how much time you have to stay with us to answer the question. How much
No, I can
time?
stay. No problem. Sorry,
Okay. Sorry. So we have a question from Sassawat. How does PEG compare with zoeterionic polymers in antifouling applications?
That's a good question. So I think the general consensus is zoeterionic polymers are slightly better, mostly because they combine more water molecules. Because they have not only just the lone pair from the ethers and the PEG, but you have these two charges, which can better structure water. So you can sort of extend the structuring further out into the water domain and trap more water in that cage, which makes it harder for proteins to get in. But again, I think that really depends on how you functionalize each of these, right? So depending what other functional groups or how the polymers are connected together or connected to your surface, I think that could have a huge variation as well in the properties you get.
Thank you so much. The next question is about byproducts of hydrazine and aldehyde. Are they toxic? I mean,
that's a good question. So I mean, when I say hydrazide, people think hydrazine. So really, you think, are we trying to blow up patients? The answer is obviously no. So we functionalize the hydrazide using adipic acid dihydrazide, which is actually a relatively safe molecule. And the degradation products, the hydrozides don't really react with anything else in the body directly. You need another activator to do so. Aldehydes, you're right. As I said in the presentation, you can form shift bases with proteins. If you have too many aldehydes, we do know that can cause a problem. So what we always do is we work in an excess of hydrazide polymer. So there's very few, if any, free aldehydes. And also, as I mentioned, incorporating more ketone functionalities inside these polymers. It's really something we're pursuing now. The ketone monomer is very difficult to make. It's not commercially available. We make it ourselves. So that's definitely a translational barrier to that. But we see significantly lower issues. We can work in excess ketones, and it doesn't cause a problem with inflammation in that case. So I hope that I answered that question, but I would say it's not that there are no concerns, but there are ways that we can mediate those concerns by properly designing the gels.
Perfect. Thank you so much. Murais has a question about printing, probably fresh printing. He's asking if in solution printing with cells, do you achieve good cell viability afterwards?
Yes. So the images I showed you are actually live dead assay stains where we like to check the viability afterwards. So we can actually maintain very high viability. And again, the reason we think this is we know that we've done a lot of injectable gels with cells without printing. We know that these materials are pretty non-toxic. And because the viscosity of the polymers we're using to extrude is basically like water. They're extremely low viscosity. So you don't get nearly the same shear forces you get with cells as you do with printing, even with alginate or shear thinning hydrogels. So I think that's the reason we're getting such good cell viability, particularly with that premixed solution where it's just a single syringe into the bath. We're seeing extremely good cell viability. And so that's our hypothesis, but it's something we're definitely still working on. We need to validate.
Perfect. Megan is asking how high is the cell seating density in this printing application that you show? When you work with this high seating density, do you get any inhibition of the gelation?
That's a great question. So the really dense one that I showed you with a lot of green dots, that was actually 10 million cells per milliliter we printed. And again, I won't say there is no inhibition of the gelation. That would not be true. But again, it's something we can adapt the formulation to account for. So if we just add more polymer or we increase the degree of functionalization, we tend to prefer to add more polymer as that tends to be better for cytotoxicity. If you create really highly aldehyde or functionalized polymer in particular, you can at some point run into issues. So we just have to increase the concentration of the polymer a little bit to compensate for the competing interactions we might get with cells. And then we can print gels. Again, the gel I showed you, I think, was 14 days, and it's still intact. You can still see the structure. And that's something, again, we can control with polymer density. So again, that's a good question. You're absolutely right. It's not a plug and play, but it's something that we can adapt the formulation to address.
Perfect. Thank you so much. Nureddin, one of our previous speaker, asking about how the starch degrades by tissue and which enzyme contribute to that. Is it inside the GI tract or, I mean, any other tissue, and what kind of enzyme?
Great question. I would be lying if I said I knew the answer to that so far. We do know that it does degrade. We see evidence for that, but the mechanism of that degradation, I don't know. I mean, starch usually is amylase, right? But then again, that's more digestive enzyme. It's not something we have a ton of just in tissues. Our guess is it's just kind of a general oxidative degradation over time as many carbohydrates break down. But that's a great question. I would like to have a better answer, but I don't at the moment.
Yeah, thank you so much. The last question from our audience, no, not the last, because we have one in the chat. Mohammed Reza is asking, is the VetZen applicable or applicable on the wound bed instead of the traditional hydrogel? Is there any benefit?
On a wound bed? It's a good question. I mean, that particular formulation is really, really non-adhesive. Which is exactly what we want inside an E, because you don't want it sticking to the surrounding tissues for lubrication. But for a wound healing application, I don't know whether that is necessarily what you want, depending on what your aim is. So we are looking at, for wound healing, a zodiac polymer in a different context, but one that's sort of a removable bandage where we include it in a Band-Aid that we then apply. So the injectability in that case maybe is not as useful. So I mean, if you have an idea, I'd be happy to chat with you about it. It's not something we felt was useful, but I guess I can't ignore the first point I gave on my second last slide, that sometimes the things we think are useless are actually what people are interested in. So happy to engage in that.
Perfect. So there is a question in the chat box also. Are these hydrogels can be used, I mean, can these hydrogel be used in transmucosal drug delivery system?
Yeah, so I mean, the intranasal system is essentially a transmucosal drug delivery system, right? So, we're quite interested in trying to look at different mucosal interfaces in the body and seeing whether we can deliver appropriate pharmaceuticals for those situations. So, you scooped me, but thank you. You're absolutely right. I think it's an interesting thing to look at.
Thank you so much. And then I have two questions. One about, as a user of these, because you said that we can buy this product from Millipore or Sigma. When we buy, we got it as a powder or as a gel?
It comes as a solution. So it's a 25% solution in water. Yeah.
Are they or is it sterilized, or how do you sterilize them?
It's not sterilized, but again, it's a really low viscosity polymer solution that, with the molecular weights are low. So they're in the order of 15 to 20 kilodaltons. So it's very easy to do a sterile filter it. We didn't sterile filter it in the product, and Sigma didn't want us to, because it's an additional cost. They would charge more, and not everyone needs it sterile filtered, right? So we have instructions. So there's a certificate analysis and there's use instructions you can download about how we suggest you use the product and filter it if you want to. I mean, honestly, if you're really interested in the polymer, talk to me and don't buy it from Sigma, because you will be paying much more than I will charge you. But
I was trying to advertise for other users.
Well, just tell all your friends who have more money than they know what to do with in research to buy it from Sigma. But if we're happy to collaborate. But yeah, it's very easy to sterile filter, so that's what we do.
But that's a perfect model. I really like the model that Millipore or Sigma is doing in terms of, you don't transfer your IP, and then you get the feedback from the customers too. This is very great. And you can track which kind of application people are using your hydrogel. This is really great.
It's unfortunate they don't give you specific, so we know what universities bought it. We don't know which labs at those universities bought it. So I really wish we had that information too, but at least we know where to look. And then the companies, you get the information directly. So it's been quite interesting, actually. So yeah.
Perfect.
It's really good.
Thank you so much. I don't have any other question. I don't know if Mohsen has some specific question today.
No, well, thank you very much, Professor Hoare, for your very interesting and inspiring talk. Well, Human asked the questions that I also had, so I think with that, I have no more questions from us. Thank you very much again.
I just want to encourage all the audience again to hang in there for next week. We have Professor Lodak from University of Alberta, who's talking about biomaterials for siRNA delivery for cancer therapy. And then we continue this seminar series to the end of the year, and then hopefully we will have it after the new year too. Thank you so much. And as you can see, Professor Hoare, we have people thanks you for your presentation and the great talk. And hopefully we will see each other soon in person.
I hope so. Thank you very much.
Yes, thank you. Have a great day. Bye-bye.
Bye.
Bye.